Alhamdulillah, how are you?
Alhamdulillah. Hidayah, how are you?
Alhamdulillah, Prof. Hopefully everything goes smooth and well, inshallah.
Bismillahirrahmanirrahim. Assalamualaikum to everyone. Welcome to our special CME. We bring this special CME just for everyone with regard to hot topic at the moment, COVID-19 vaccine, what we know so far. So the event is organized by Kuliah of Medicine in RIUM. And we have two distinct speakers. Before that, my name is Jamaluddin Abdul Rahman. I'm currently the Deputy Dean Postgraduate and Research in Kuliah of Medicine. And today we have two speakers, none others than our professors and also medical microbiologist, immunologist, Prof. Imad Mustafa, who is also the head department of basic medical sciences in our kuliah. And a bit background with regard to Prof. Imad, he had his medical degree from Baghdad and further his PhD in Sheffield. He is currently members of European Society of Microbiology and Infectious Diseases, as well as Australian Society of Microbiology. The second, Prof. Imad will be talking about types and their potential hazards, in terms of type of vaccines and their potential hazards. And also we have Dr. Hidayah, who is currently assistant professor at the Department of Pathology and Laboratory Medicine, Kuliah of Medicine. She is a clinical microbiologist, also a coordinator of Infection and Control Unit, SASMEC, Sultan Ahmad Shah Medical Center. She obtained her master in from UKM, and she's actually our graduate. Saya pernah ajar ke Hidayah?
Yes, both of you, Prof.
Oh, ya ke? Ya ke? I feel like I would like to deny that lah. Macam I feel like too old.
I'm from
okay, nevertheless. Okay, Daya. It is great to have both of you. Now the teacher and the students combined. We try to talk about vaccines, COVID-19 vaccines. And for the audience, you can ask after Prof. Imad give his talk, and then Hidayah give his talk, and then I will see the chat box with any questions that you would like to ask. That will be easier. So you mention your name, your full name, where you are, and then where you're coming from, and then what would be the questions? Sweet and simple kind of questions. Your name, full name, and then where you're coming from, because I also noticed that we also have guests from other than Kuliah of Medicine, from other than UAE. So then they mention your questions. All right. I may not be able to answer to pick all the questions. And a bit about the etiquette of our meeting today, our conference today, is that please guys mute your mic, and then switch off the video except for the speaker when they are speaking. And if you have any question, please submit using the chat box. So I think Prof. Imad, the screen is yours. The floor, the screen is yours. So I will stop my share now. You can share your
Okay, thank you, Prof. Jamal. So you can see my screen?
Yes.
Yeah, Prof.
Yes. Okay. Okay, Assalamu alaikum wa rahmatullahi wa barakatuh. So these are the contents of my talk today. So I'll give you a brief introduction on SARS-CoV-2 structure, especially regarding the major viral proteins. which are important for vaccine development. And then a little bit on epidemiology in relation to vaccination. And then I'll talk about immunological correlates of protection in case of COVID-19 infections and probably vaccination. And then I'll talk a little bit on 4 main types of COVID-19 vaccines, and then the impact of mutant viral variants on immunity to COVID-19 vaccines. So this is a very well publicized diagram of the SARS-CoV-2 virus. And these are the spikes, the one that you see here, the blobs sticking out from the surface of this sphere. These are the S protein. This is the S protein, which has 2 subunits, the S1 and S2. The S1 is important for binding to the receptor, which is ACE2 molecule present on cell membrane of various types of cells that can be infected by the virus. And then there are some other proteins which are important for the structure of the virus. And these are the matrix protein, or membrane protein referred to nowadays, membrane protein. And also we have the E protein present again on the surface of the virus. In addition to these 3 proteins, there are other proteins such as the nucleoprotein, which is you cannot see here because this virus is still intact. So it is inside. The nucleoprotein is the one which is associated with the genome of the virus. And there are many actually, about 16 non-structural proteins, which are also important for the replication of the virus, plus accessory proteins. So this is quite actually a big virus regarding its genome. It has about 30K bases in its genome. So it is quite a big virus. This is from an article written by these scientists, Chong and Jones, saying that the COVID-19 is a complex of medical conditions. And the ultimate causative agent is not the virus in isolation, it's not the virus alone, but a complex with particular social factors, which they dubbed as the 4 horsemen of destruction, actually, the 4 horsemen of destruction, which are number 1, overpopulation, globalization, hyperconnectivity between people, and the extreme centralization and increasing fragility of supply chains. And these 4 factors, detrimental factors, might operate not only on COVID-19, but in any further epidemics or pandemics that might arise, la samahullah. So regarding the impact of vaccination on the epidemiology of COVID-19, we know that the COVID-19, most infections are mild infections or even subclinical infections. 15% are quite symptomatic infections. Some of them might be quite severe. And only 5% are critical infections that might require intensive care admission. So if we consider that in order to achieve a herd immunity regarding COVID-19 to prevent community spread of the virus, the herd immunity level required is about 70%. So in Malaysia, with a population of about 33 million or 34 million, this means it is over, this 70% will amount to about 20 million or more. So meaning that if we wait for herd immunity to occur, to stop the spread of the virus in the community, it means that among these 20 plus millions, 5% of them will have quite severe infections that require intensive care admission. And 5% is about 1 million actually, isn't it? You can correct me if I am wrong about the calculations, is about 1 million. And if we consider that the mortality rate is about 0.5 to 1%, it is 1% mortality among all the infected persons, then about 100 to 200,000 will succumb to the infection before we can achieve this 70% level of herd immunity. So this is one important reason why vaccination is quite important. Mass vaccination is quite important. So this slide just shows the effect of vaccination and on the spread of the virus. This is again well publicized diagram of how the virus spread in non-immune or non-vaccinated population, and how the spread is halted in case of vaccinated population. Now, people started work on the development of COVID-19 vaccine as soon as the whole sequence, genomic sequence of the virus was published on 11 January 2020 from China. And then pharmaceutical companies, and even before the COVID-19 pandemic, they were working on vaccine development using new technologies. And this actually, so this utilization of the pre-pandemic vaccine knowledge that they have acquired and experience led to the speedy development of the current COVID-19 vaccines. So what were the technologies that led to this rapid development? Number one is reverse vaccinology, or in silico analysis of the viral genomes to find out what are the antigens and epitopes which are associated with, most probably associated with immunity to the virus infection. So reverse vaccinology is defined as the process of antigen discovery starting from the genome information. And the genome information we already had from China since the beginning of the pandemic in January. So that's why people started working on developing these vaccines. And actually, reverse vaccinology was applied before that, as probably in the early parts of the 21st century, and mainly used for development, first used for development of vaccine for Neisseria meningitis, group B, which is quite very difficult to get a vaccine for this type of Neisseria meningitis, the group B, because the capsular antigen vaccine is not effective. The conjugated capsular antigen vaccine is also not effective. So they had to develop a new type of vaccine using reverse vaccinology, and this vaccine consisted of actually certain peptides that give protective immune responses in vaccinees. So again, the COVID-19 vaccine accelerated development is due to the new ways people have developed these vaccines, working in parallel or overlapping. So they do not have to complete one phase to go to the second phase, but we have here overlapping phases and for the development of the vaccine. So this is one, again, important factor in accelerating the development of the vaccine. Instead of needing about 10 years or so for developing the vaccine by the conventional ways, they managed to develop the vaccine even within 1 year or two. So Immunogen used to develop viral vaccines in general, not only for COVID, include attenuated live vaccines, meaning a virus that has been made weak enough to might cause an infection, should cause an infection, but not lead to disease, not symptomatic disease, and no pathology is induced. And then there is the whole inactivated virus vaccines, meaning you kill the virus by either chemical means using, for example, formalin or beta-propiolactone, which is a chemical that is used since long time for inactivating microorganisms, or using UV lights, or a combination of these inactivating factors. So you will have, at the end, you will have an inactivated vaccine. Now, what's the difference between these two? Live attenuated vaccines, of course, they can self-amplify when given to the host because they are live vaccines. So the microorganism can multiply, and the antigen load will amplify again in the host. So you don't need to give high dose. You give usually much smaller dose than you give in case of inactivated vaccines. Also, inactivated vaccines might be weaker immunogens, so they require adjuvants to enhance immunity. And then we have the protein subunit vaccines where you give certain viral proteins or antigens that are known to be associated or induce protective immunity in the vaccinee. And these are, again, weakly immunogenic and might need adjuvants to be given together so that the immune response is enhanced. And then we have recombinant vaccines. I don't want to talk about now, such as recombinant vaccinia virus vaccine, where you introduce genes that code for protective antigen responses, protective antibody responses from various pathogens. So you can include actually several proteins from several viruses or other microorganisms. Then we have the synthetic peptide vaccines, where you, if you know the sequence, the amino acid sequence of the peptide or proteins that induce protective immunity, can synthesize these peptides. But again, these peptides are poorly immunogenic, so they require adjuvants to increase their immunogenicity. And then we have vector vaccines. Vector vaccines, which are being used nowadays and for COVID-19, where you introduce a part of the viral genome coding for an immunogenic antigen, an antigen that induces protective immunity, and another virus, and another viral particle, which can be replicating or non-replicating. And then this virus can cause infection and induce protective immune responses. And one of the most commonly used are adenoviruses, human adenoviruses or monkey adenoviruses. And then we have the nucleic acid vaccines, again developed recently for the first time for the COVID-19. And these can be either DNA or RNA containing. The DNA also code for a certain, for a gene, code for a protein, contains a gene that codes for an antigen of the virus that induces protective immunity. And this can be present associated with a plasmid and then given by injection together with an electric current has to be introduced so that you induce electroporation so that the DNA can penetrate the cell. And then we have the messenger RNA vaccine. And the messenger RNA we'll talk about later. And both of them can induce actually protective immune response, strong protective immune response in the host. The messenger, both of them will utilize the host cell to amplify the immunogenic viral proteins. So these vaccines are newly developed vaccines. They have not been used before for human vaccination. They are known, but they have not been used before in human vaccines. So this is again a diagram showing the types of vaccines. This is vaccine from whole virus, which can be, as we said before, live attenuated or inactivated. So attenuated live virus can amplify when given into the host. and can, if taken up by professional antigen presenting cells such as dendritic cells or macrophages, they can present these antigens to other immune cells, T cells, and B cells to induce both humoral and T cell responses. The inactivated vaccines usually can induce, more commonly induce strong humoral responses, but poor actually T cell responses. So this is one drawback or differential point between live attenuated and killed vaccines. Then we have the protein-based vaccines, which include the protein subunits and also virus-like particles. So protein subunits such as the hepatitis B vaccine, which contain the hepatitis B surface antigen, which is synthesized in eukaryotic cells and actually yeast cells, and then purified and given as a vaccine. The hepatitis B surface antigen is given a vaccine here. It is the spike protein for COVID-19. Of course, you use the spike protein, which contains the antigen binding domain of the virus. And this can induce neutralizing antibody responses. In addition, people claim that they also induce T cell mediated immune responses. And then we have the virus-like particles. An example of virus-like particles, these particles look like a virus particle, but they are actually not viruses. These are lipid vesicles with the exogenous protein studied on the surface. So they look like a virus particle. They can be uptaken by professional antigen presenting cells and induce good immune responses. And example of these are human papillomaviral vaccines that are known now for some time. Then we have the viral vector vaccines, can be a replicating vector or non-replicating vector. And the most commonly used vector, as I have mentioned before, are adenoviruses, which can be human adenoviruses or that are made non-replicating, inactivated, or monkey adenoviruses that do not cause disease in humans. Then we have the nucleic acid vaccine that we have mentioned before, which include DNA vaccines. The DNA consists actually, the DNA they include introduced into a plasmid. Plasmid genome is also a DNA genome. And this piece of DNA is chosen from a sequence that code for the immunogenic viral proteins, such as in case of COVID-19, it is the S protein. Also, we can use messenger RNA to be introduced, and then the messenger RNA will be translated directly by the host cell ribosomes, and then modified in endoplasmic reticulum and Golgi apparatus. So we will end up with a viral protein, immunogenic viral protein, a viral protein that induces protective immune responses in the host. Now, people are afraid of DNA plasmid vaccine because DNA, they are afraid has to enter the nucleus, not only the cytoplasm. The DNA plasmid vaccines, they have to penetrate not only the cytoplasm, they have to penetrate the nucleus of the cell, of the host cell. But these plasmid vaccines, DNA plasmid vaccines, do not integrate into the host cell DNA. So there is no fear of integration and then causing disruption or mutations in the host DNA. Regarding the messenger RNA vaccine, the messenger RNA has only to penetrate the plasma membrane of the cell, and then the released messenger RNA will be translated by the host cell ribosomes, and then, as we said, modified by the endoplasmic reticulum and Golgi apparatus, and then expressed either excreted extracellularly or expressed on the surface, and can stimulate immune responses, both humoral and cellular immune responses, T-cell mediated immune responses. So the DNA plasmid vaccine, actually, the DNA plasmid that is introduced, as we said, will not integrate into the host cell DNA, and it will not remain for very long time. It will be destroyed after some time. Also, the messenger RNA that is introduced will be degraded after a period of time. How long? It might be hours, or it might be a few days. Now, immunological correlates of protection for viral infection, especially here for the COVID-19, should be balanced immune responses, innate immune response that will induce the two important types of interferons, interferon 1 and 3. The type 3 interferon is a more recently identified interferon. We knew before interferon 1, which has the types alpha and beta, and then interferon 2, which is the gamma interferon, but the type 3 is the lambda interferon, which there are 4 subtypes of it. So these are protective interferons if they are induced early, in the infection. But if they are induced in excess, late in the infection, they might be detrimental. They can enhance inflammatory reactions. Then we have adaptive T-cell immune responses, which include, of course, the CD4. And the CD4 T helper cells, we have T helper 1 and 2, and some others, several others, but these are the ones that are most important now. The T helper 1 is known to induce protective cell-mediated immune response. But T helper 2 responses and T helper 17 responses are not considered as favorable because they can induce allergic reaction. The T helper 2 can lead to allergic reaction by inducing switching from IgM to IgE production, which is involved in allergic reactions, and T helper 17, which can produce pro-inflammatory cytokines, and T helper 17 is now known to be involved in many autoimmune diseases. So these are unfavorable. So the immune response can be directed, the adaptive T-cell immune response directed to T helper 1. This will be beneficial. And also the CD8, which is the cytotoxic T-cell response, is also effective in combating, in killing virus-infected cells before they can spread the infection. And then we have the adaptive humoral antibody responses. So we have T-cell responses and we have antibody responses. Both of them are part of the adaptive immune responses. Now, the important antibody responses required to combat, stop the viral infection are neutralizing antibodies. So neutralizing antibodies in case of COVID-19 are directed against the S protein of the virus, especially the S1, which contain the antigen binding site, the receptor binding site, which is the ACE2. So antibodies to this region, to the S1, especially to the receptor binding domain, then this can be protective and neutralize the infectivity of the virus. That's why it's called the neutralizing antibody, neutralizing the infectivity of the virus. Neutralizing antibodies can be also against toxins, and then they will be called antitoxins. Anyhow, we don't have antitoxins here, so we are talking about infectivity. So these are neutralizing antibodies that will protect against the infection of viral infection of cells. Unfavorable responses here include non-neutralizing, enhancing antibodies. Enhancing antibodies has been known since long time for some flaviviruses, such as dengue virus, and they are probably associated with the more severe forms of dengue, the dengue shock syndrome and hemorrhagic fever. And they are also known to occur more recently in Zika virus, which is again a flavivirus. Also, development of excess antigen antibody immune complexes is not a good or favorable humoral response, antibody response, because it might lead to type 3 hypersensitivity reactions. So the immune responses to the COVID SARS-CoV-2 infections, SARS-CoV-2 can induce imbalanced host immune responses, such as early interferon 1 and 3 levels inhibit, actually, attenuated interferon 1 and 3 levels, early infection, but they become high later on. And also increase the level of pro-inflammatory cytokines. So SARS-CoV-2 is known to suppress interferon 1 and probably 3 production early in the infection. And these interferons are very important antiviral proteins. Regarding adaptive immune response, as we said, CD4, T helper 1, and cytotoxic T cells are important. And these the cytotoxic T cells and the T helper cells can recognize not only the S protein, but many other viral proteins, structural viral proteins, and even non-structural viral proteins that might be associated with immunity. So this is a good thing about vaccines that can induce both humoral and T cell responses. The antibody responses in case of COVID-19 infections are mainly the important ones for protection are the neutralizing antibodies, especially the ones that bind to the receptor binding domain. Receptor binding domain is present on the S1 subunit of the spike of the virus. And it is important for neutralizing the infectivity of the virus. And these antibodies are mainly IgG with some or weaker IgM and IgA responses have been noted in case of COVID-19. Now, what about waning immune responses and reinfection? Reinfections actually have been, few reinfections have been documented, but reinfections, even if they occur, they are supposed to be cause mild infections rather than severe infections. If they cause mild infections, then they will boost the immune response. They will induce a secondary immune response that will boost the immunity of the host. But, however, people have noted that some people with second infections might develop severe disease. We don't know exactly why. Now, waning, more recently on 5th of November 2020, the Oxford University Hospital Group have found that COVID-19 antibodies decline to have their original level within 3 months after infection, decline within 3 months to have their original value after infection, after an acute infection. What about the impact of this on vaccination? This probably might, if it is confirmed, it means that you have to repeat the vaccination, revaccinate within a period of 6 months or 1 year, probably. You have to repeat the vaccination, especially in the old age group where the immune response might be weaker. So you have to revaccinate after 1 year or 2 years, we don't know yet, so we have to wait and see. Vaccine efficacy, this is just the formula for how to derive the vaccine efficacy, so you can have a look at it. Current COVID-19 vaccines, there are currently 43 vaccines in phase 1, 20 in phase 2, 18 in phase 3, 6 others approved 2 till now, and more probably will be approved soon. And 1 is abandoned, such as the vaccine developed in Australia, because it induces false positive HIV test response. So it has been abandoned. So there are 63 vaccines in clinical trials in humans, and 18 in the final stages of testing, and 85 in preclinical development. These are the stages in development of vaccines. So till now we have reached only phase 3 clinical trials, and these phase 3 clinical trials have showed that the tested vaccines, such as the ones from Moderna and Pfizer, are both effective and safe, relatively safe, although there are many reports of quite systemic reactions, moderately severe systemic reactions in some individuals, but they are not serious. So serious reaction to viral vaccines include local reactions such as pain at the site, and systemic reactions such as fever, muscle aches, and so on. Phase 4, which is the post-marketing surveillance, we have not arrived yet. So after mass vaccination, probably we can look up for the phase 4 results. So this is just a repetition of what we have mentioned before. So these are the 4 main types of current COVID-19 vaccines. We have the messenger RNA vaccines, vector vaccines, protein subunit vaccines, whole killed vaccines. And probably we have to add the DNA vaccines here. So messenger RNA vaccines are being developed by Pfizer, Moderna, and you have to give 2 doses of the vaccine. So 1 dose is not quite enough to induce a protective immune response. So you have to give 2 doses about 20 days apart. And then there will be a protective immune response about 1 week or so after the 2nd dose. So the total is about 4 weeks or 1 month. Then we have the vector vaccines, where they use mainly adenovirus vectors, either the human adenovirus or the chimpanzee or monkey adenoviruses. And these have been developed by AstraZeneca, Janssen, Sputnik, and Russia. And the doses are again range between 1 and 2 to induce a protective immune response. Then we have the protein subunit vaccines developed by Novavax, Sanofi, and the doses are again 1 to 2, depending on the type of the vaccine. And then the whole killed vaccines are also being developed by Sinovac. And there are other vaccines are actually in development. So the leading vaccines include these: Pfizer, BioNTech, messenger RNA vaccine, Moderna messenger RNA, CanSino adenoviral vector vaccines, Jamalia adenovirus from Russia, Johnson & Johnson adenoviral vector vaccine, Oxford AstraZeneca adenoviral vaccines, and Vector Institute protein vaccine, Novavax protein vaccine, and the Sinopharm Beijing and Sinopharm Wuhan vaccines, both inactivated vaccines, and a Sinovac vaccine, which is also inactivated. Now, we all have heard about the recent widespread of a new mutant of coronavirus, and especially in southeast UK, in London in particular. And this new variant is in the media, people have said that this is a super spreader viral strain of the virus. Actually, these claims are not substantiated. The virus, first of all, it is not easy to find out whether a virus spreads more easily than the one before, than the non-mutant virus before, because there are many confounding factors, so this needs time to be confirmed. And the disease produced is not more severe than the one than the other ones caused by the other viruses, virus strains, if you can call them. Regarding naming these mutants as strains, a viral strain should have some new biological properties which are not present in the previous non-mutated strain. And this does not seem to be true for COVID-19, for the SARS-CoV-2 viruses. There are mutations, of course. RNA viruses in general mutate at a much more rapid rate, many of them, not all, of course, than the DNA viruses, because they do not have the proofreading exonuclease activity. But the coronavirus has exonuclease activity and has some proofreading ability. So the mutation rate in in SARS-CoV-2 is much less than, for example, in influenza viruses or in HIV viruses, which are known to mutate at a relatively high rate. So the impact of this new variant of SARS-CoV-2, people are watching out for the properties of and the spread of this virus and the severity of the infection caused by this virus. But till now, there is no firm evidence that this mutant spread more easily or cause more severe disease. And people think till now that this virus variant, this mutant variant of SARS-CoV-2 will not affect the vaccination. I mean, it can, vaccines can prevent or interfere with infection caused by this mutant till now. But if more mutations occur, especially the mutation that affect the the receptor binding domain of the virus, then you might have some other talk about this. But till now, it seems that the mutations, especially this one mutation actually, from N501 to Y, N and Y, these represent the mutations that lead to change of the amino acid from one type to another. N, I think it stands for asparagine, Y for, I forgot what Y stands for, but this is, these are, and the 501 represent the site at which this change occurred, this mutation has occurred at site 501. And most mutations actually, viruses, as we said, especially RNA viruses, undergo a lot of mutations, but most of these mutations are neutral mutations. They do not affect the biological behavior of the virus, and they do not affect also the antigenicity of the virus. So most of these mutations are neutral mutations. Of course, lethal mutations, if they occur, you cannot find them because the virus will not be able to replicate and cause infection, so it will not be detected. But on the other side, some mutations can cause attenuation, can make the virus less virulent. So, and this has been noted for some of these mutants. So some mutants that have arisen till now, some of them have lower pathogenicity than the non-mutant viruses. So that's all from me. Thank you very much. Wassalamu alaikum.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Prof. Imad. I believe I do have few questions, but we stick to our agreement. We'll listen to Dr. Hidayah. For those who just joined us, we have two distinguished speakers. We have Prof. Imad, who is the medical virologist, and we also have Dr. Hidayah, who is our clinical microbiologist at Kuliah of Medicine, as well as in Sultan Ahmad Shah Medical Center. So for the second talk, I would like to invite Dr. Hidayah to present your talk. You can share. Okay, thank you. You're mute, Hidayah. Wait, let me, yeah. Okay.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim.
Salam.
Thank you, Prof. Jamal. Thank you, Prof. Imad. Thank you again to the organizing committee for inviting me to speak at this CME. I will try my best to deliver, inshallah. Okay. Next slide. This is the content of my presentation. I'll begin with a brief introduction on vaccine development and safety monitoring. This part actually Prof. Imad had actually touched a little bit on his presentation. And afterwards, I will explain briefly on adverse event following immunization. What are the common local versus systemic events, systemic reaction, minor reaction versus serious reaction, and a little bit on the COVID-19 vaccine safety profile. Obviously, we at this moment of time, we only have the short-term safety profile rather than long-term. And the types of COVID-19 vaccine that have this safety data information only limited for the two approved so far, which is the Pfizer BioNTech vaccine and Moderna vaccine. And last but not least, conclusion. By now, I think all of you know that vaccine is very critical in terms of disease prevention at a population level. It protects the individual and it prevents transmission within the community. And it is the utmost aim is to have an access to an effective and safe vaccine. In terms of COVID, an unprecedented political, financial, and populist pressure to find a suitable COVID vaccine risks us to miss this aim of getting an access to effective and safe vaccine. And worst come to worst, it can damage the public confidence. So to safeguard this aim, WHO have actually set certain standard, the certain minimum standard that have to be made for any COVID-19 vaccine that are to be manufactured. The vaccine should at least have efficacy of at least 50%, and it should shows a good temperature stability and potentially adequate for a rapid scale up in terms of production and manufacturing, and it must have a good safety profile. Okay, this image basically summarize the three important phases in a clinical trial or clinical developmental stage of a vaccine. And at least the FDA have ensured us that they will only license vaccines that are safe and effective enough where the benefits outweigh the risks. Okay. These are the three main international bodies that basically governs program evolving around vaccine. WHO, for example, is very committed to accelerate vaccine development while maintaining the highest safety standards. They won't, they cannot afford to jeopardize the safety standards of vaccine, especially those candidate vaccine that are to be used for general public. Okay, what drives the urgent need of a COVID-19 vaccine? In my personal opinion, I think that it is due to the intense burden on the healthcare system. Our healthcare system, any affected healthcare system could not cope with intense spread of COVID-19 within the community. And another thing is that is due to the massive impact in economy. Not so much on the mortality, because the overall mortality of COVID-19 infection so far is only at 3.4%. We're not talking about those who are unwell, those who are with uncontrolled severe medical conditions, where the chance of them getting a fatal disease is about 50%. So in the presence of unprecedented financial investment and scientific collaboration, really change how vaccines are being developed. I think Prof. Imad just now have also mentioned about the sequential step of vaccine development have been compacted, a few steps occurring in parallel while they still maintain a strict clinical and safety standards. Okay. As for the US is concerned, they have this the Food and Drug Administration, the FDA, to regulate vaccines approval. I think each and every individual country have their own regulating bodies. For example, in Europe, they have a European Commission to look into this and the rollout of vaccine. Malaysia also we got. Okay, I will explain it a little bit. And most importantly, upon rollout of this vaccine, these organizations are very committed to conduct a robust post-licensure monitoring of vaccine safety. We do have also here in Malaysia, we have a special organization to monitor this. In early December 2020, Pfizer-BioNTech vaccine is the first to receive EUA from at least 7 different countries worldwide, which is Canada, US, Bahrain, UK, Saudi Arabia, Mexico, and Singapore. It is basically a novel vaccine with unknown long-term side effects. You don't know what is the long-term side effects, and this sparks anxiety and to a certain degree hesitancy among the public. I did attach a link here in my presentation, so you can watch it later on. Basically, it's how FDA are trying to convince the public that they are doing the correct process of developing a good vaccine. In terms of adverse event following immunization, there's no such thing as a perfect vaccine. Every effective vaccine may produce some undesirable side effects. And most of the time, majority of the events are actually not due to the vaccine itself. It may be simply due to coincidental events. Others are due to error, either in human error or the program error. And this adverse event can be unpredictable at a certain extent. Okay, what is meant by serious adverse event? Serious adverse event usually involve death, hospitalization or prolongation of existing hospitalization, persistent or significant disability or incapacity, resulting in congenital anomaly and even life-threatening. Okay, this is a few selected childhood vaccine that I've included here just to show you that following a vaccination, local reaction and systemic reactions is common. And among the common local reactions are the pain at injection site, swelling, and redness. Important systemic reactions include fever, fatigue, malaise, and so on. Okay, it's not. It is something that indicates that the vaccine component is eliciting a response inside the vaccine recipients, just like a marker like that. Okay, these are the example of severe vaccine reaction. I placed it here, for example, measles. Measles for the vaccine vaccine recipients may develop into anaphylaxis, but the frequency is about 1 in 100,000, not that frequent. And upon receiving the vaccine, the reaction is usually immediate. It's if for anaphylaxis. And all other sorts of severe vaccine reactions. The following subsequent slides, I will keep on giving a few examples with regards to the side effects of few established vaccine. The first example is the Rotashield. Rotashield is the first licensed rotavirus vaccine. Upon enrollment in year 1998, there are increased incidence of intersusception among the vaccine recipients, which resulted in the withdrawal of Rotashield from the market a year later. This first example. Second example is the yellow fever vaccine. Upon enrollment, there have been more than 60 cases worldwide have been reported to CDC, documenting this neurological development of neurologic disease. Okay, yellow fever vaccine is a live attenuated vaccine. One of the complications of live attenuated vaccine is that the vaccine strain itself has gained its strength and caused disease in the recipients. This is one of the known side effects of a live vaccine. Smallpox vaccine is also a form of live vaccine. Okay, it can cause disease in the vaccine recipient and can eventually lead to death. There have been 1 to 4 deaths per million vaccinated person have been reported. Okay, Dengvaxia. Dengvaxia is the first FDA-approved vaccine for the prevention of dengue, at least in the endemic region. It's the first FDA-approved. So upon approval of Dengvaxia, everyone got very excited, including us in Malaysia. Dengvaxia release, it has got its first license for distribution in May 2019. The initial uptake for Dengvaxia is phenomenal, where countries like Brazil, a few other African countries, even Asian countries like Philippines, include Dengvaxia into their national vaccination program. Dengvaxia do reach our shore, and MOH did participate in a phase 3 trial by giving the Sanofi Pasteur, the manufacturer, a conditional approval for local distribution. The licensing was based on the data collected 1 year after administering the third dose in phase 3 trial. As time goes by, data obtained 5 years later, post-immunization results showed occurrence of vaccine-enhanced disease in apparently in the age group that is being studied. This is the phenomenon where Prof. Imad just now did mention briefly, where when a group of recipients who have not been exposed to that disease in the first place, given a vaccine exposed to the dengue virus, will later develop a severe disease. It has a very detrimental consequence where it ends up in Philippines, for example, the vaccine was withdrawn from their mass vaccination program and even banned the use of this product in their country. This is the implication. It turned out the public confidence with regards to the safe and effective vaccine. Okay, what about COVID-19 vaccine COVID-19 vaccine safety profile? In November this year, our government have signed the commitment agreement to purchase Pfizer-BioNTech vaccine. Local use will be subjected to our local vaccine regulatory body, which is the National Pharmaceutical Regulatory Agency. The approval process are currently being conducted. And the usual process for approval can be ranged from 90 days to 120 days. And the government is really committed to shorten up this duration to 90 days. And it is projected that this vaccine will reach our shore in the first quarter of 2021, which is most likely in February. That involves 12.8 million doses, aiming for at least 20% of national coverage, which equals to 6.4 million vaccine recipients. But latest information that we got that the government have increased the coverage of vaccine, the target of coverage vaccine from 20% to 80%, and they have spent billions to obtain the vaccine. And our government decision to purchase this vaccine will make based on the favorable interim clinical efficacy and safety report. This is just published on the 10th of December in the New England Journal of Medicine. You can take a look at the full article at the link provided. It's basically looking into BNT162b2 mRNA COVID-19 vaccine. Okay, it is a multinational study involving mainly the citizens of United States of America, aged more than 16 years old, and they have been divided into two groups, 16 to 55 years old, and another group more than 55 years old. Previous COVID-19 infection, immunocompromised, and pregnant women were excluded. 2 doses, 21 days apart. The endpoints, it measures efficacy and safety. A total of 21,270 were vaccinated in comparison to the another equal number of placebo group. Okay, I'm not going to talk in detail. In terms of efficacy for Pfizer BioNTech vaccine, it has been found out to be 95% efficacious. Okay. At the end point of the study, it has been documented that there are 8 new COVID-19 onset in vaccine group as compared to 192 new onset of COVID-19 in a placebo group. And similar efficacy was observed across all subgroup of study. There are 10 severe COVID-19 cases were observed following first dose, 9 in placebo group versus 1 in vaccine group. Okay, at any given point of time above 14 days, the occurrence of new onset of COVID-19 are always low in the group who receive BNT162b2. Okay. So efficacy data, check. For safety data, vaccine reaction were monitored at least within 7 days after first and second dose. And the severity of the reaction is categorized into mild, moderate, severe, and grade 4. Grade 4 is referring to require a visit to emergency department or require hospitalization. In short, for local reaction, most of the recipient of the vaccine complaining of mild to moderate pain at injection site. No grade 4 event occur. Okay, this is for the local events. For systemic event, fatigue, headache, chills, and muscle pain are among the common systemic event documented. Second dose usually generate a more significant systemic event as compared to the first dose. Okay, this is the younger age group for systemic event. This is for the older age group. For both local reaction and systemic reaction, the incidence is documented more in the younger age of tested population. For systemic reaction also, there's no grade 4 event documented. As for the adverse event, 50% higher incidence of adverse event occur in vaccinated group as compared to placebo. This is expected because the vaccinated groups are dealing with real vaccine. Lymphadenopathy, 0.3% in a vaccinated group. It's just showing that how robust the vaccine elicit an immune response towards the recipient. 4 serious adverse events detected in vaccinated group. 4 cases listed: shoulder injury, right axillary lymphadenopathy, paroxysmal ventricular arrhythmia, and right leg paresthesia. 4 serious adverse events. 2 deaths were reported in vaccine group. And however, both cases have been investigated and found to be not related to vaccine. And there's no COVID-19 associated death were reported. In the placebo group, there are 6 deaths. No stopping rules violated. Stopping rules are basically referring to the vaccinated group who developed severe adverse event following reaction. The threshold remains below the stop trigger. The monitoring of the study will be continued for a total of 2 years. And I think for the past few days, we have heard the news that a few of the vaccine, there are few reports associated with anaphylaxis or allergic reactions towards vaccination. This incident was not documented anywhere inside the report. Okay. This interim report mainly includes a 14 weeks of safety data on a 20,000 plus recipient of vaccine. It states that it is adequate already to observe for common adverse event. It gives an 88% probability to detect at least one adverse effect if the true incidence of the adverse effect is of 0.01%. And for the less common adverse event, of course, a larger study is required and a longer duration of study is also required. Okay, other than Pfizer BioNTech, Moderna is actually the second COVID-19 vaccine approved by the FDA. The slide is basically just showing the head-to-head comparison between the 2 vaccines. Okay, as you see here, the subjects are mainly 16 years, 18 years above. No children included within this study because it is considered age 16 and above is considered as a safe age. But Pfizer and BioNTech are committed to produce data tested on subjects aged between 12 to 15 years. Okay, there are a little bit difference in the doses of the vaccine. Okay. Storage requirement, efficacy more or less the same. So far, a few of the potential COVID-19 vaccine have an efficacy of at least 90%. And most importantly, this Pfizer and Moderna vaccine have a similar safety profile, at least at the early phase of the study or of the trial. So as a conclusion, generally, serious vaccine adverse reactions are uncommon and death due to vaccine is very rare. Short-term efficacy and safety of COVID-19 vaccines licensed for EUA is so far evidence-based, and it's just now it's a game of time, okay? And commitment and transparency from the pharmaceutical manufacturer is critical, or the vaccine manufacturer is very critical. For a better informed consent decision that requires this access to the long-term efficacy and safety monitoring data. Trust the process, at least for now, I think. And not to forget, WHO issues this statement that they warn against pandemic complacency amid vaccine rollout. So stick to your SOP, avoid the 3C and obey the 3 Ws, and eventually put a total trust in Allah. Okay, perhaps you love a thing that is bad for you, or you hate a thing which is good for you, and Allah knows while you know not. I think that's all. Thank you.
Thank you so much, Hidayah. Alhamdulillah, all the speakers already presented their talk. As mentioned by Dr. Hidayah, I think the last clause is very important to us as a Muslim. This is part of our effort to curb COVID-19, but at the same time we must put our hope lies on the hand of Allah Subhanahu wa Taala. So okay, now a Q&A session. So I would like to ask first, okay, because the moderator, I have to ask first, and then we'll open up. I also received few questions from the chat box here. Sorry, for others, I disable your mic. So if you want to ask, please use the chat box. Please put your name if you are not have the name written properly. To Prof. Imad, you did mention that mutation is generally rare, and even if it happened, it rarely affect the antigenicity of antigenic property of the virus. And then basically the tone of the presentation is as if the mutation is quite rare. But I wonder, is coronavirus COVID-19 also behave like flu, whereby we have also vaccine for flu, but you know that flu we have to take very regularly. In fact, flu vaccine can't cover all the flu virus that available. So I wonder whether vaccine for COVID-19 also will behave such as the flu vaccine. Prof. Imad? Is Prof. Imad around or already left?
Prof. Mute.
Okay.
Okay.
Okay. Thank you, Prof. Jamal. No, I did not say that mutations are rare for COVID-19. Actually, COVID-19 is, I mean, the SARS-CoV-2, which is the virus, is RNA virus. It can undergo mutation, but not as common as in case of influenza viruses or in case of HIV viruses, because it has some exonuclease proofreading ability. Mutations do commonly occur and have occurred, and people have isolated so many mutants, but they have utilized these mutations as markers for tracing infection in certain regions. So this is what I actually meant. Mutations do occur.
So if the mutation happen,
most of the mutations that happen are neutral mutations. They do not affect the infectivity, the spread of the virus, or the pathogenicity of the virus. Most of them, or even the antigenicity of the virus. Most of them are neutral mutations. But what is alarming in the last mutant that happened, occurred in UK, is that there is a mutation, one mutation, that occurred in the receptor binding site of the virus. But analysis of this mutation shows that it does not affect the antigenicity of the virus, so it will have no impact on vaccination, on vaccine protection against this mutant till now. But if further mutations occurs, then there is another talk. But at the time being, it is only this mutation. And as we said, the mutations commonly occur with this virus and other viruses, especially the RNA viruses, the non-stable ones, but these are mostly neutral mutations.
Prof. Jamal, I think...
Thank you. Okay. Sure.
I have a short note also on that. If we're to compare influenza and coronavirus, I think that might not be the case, because for influenza vaccine, the genome is actually segmented. If changes occur to the whole genome, then it is capable to produce a whole lot of new antigen, which can compromise the effectiveness of vaccine. But in terms of coronavirus, the mutation occur at single point. I don't think it's adequate to produce a whole new antigen that will alter the effectiveness of the vaccine.
Okay, thank you so much for the answers.
Scientists do convince that it won't affect for the mentioned mutation, but we see how in the future.
All right. Again, I would like to ask Prof. Imad, when you said that mRNA virus is rarely, never been used in human before, why is that?
mRNA vaccine has not been used before.
Vaccine. Sorry,
yeah, vaccine
yeah.
have not been used before. This is the first time that it's being used for this virus, for COVID-19. The reason why it has not been used, people are not so comfortable with vaccines that contain quite altered genome, because this mRNA is not natural mRNA. It is synthetic RNA that is modified RNA. But I mean, it does not last for a long time. It will enter the cytoplasm of the cell, and then it will be translated. And later on, after a certain period of time, not very well known, can be hours or days, it will be degraded. And actually, RNAs, in general, RNA molecules are unstable, not like DNA. DNA are more robust.
Okay, we have one question from Dr. Khairul Aini. She asked, does adjuvant needed for mRNA vaccine?
No, it is not required. Because the virus, I mean, the antigen is amplified in the host, in the host, and it is expressed in such a way that it is quite immunogenic. Because it can be taken up not only by muscle cells, as people think, it can be taken up by immune cells, such as dendritic cells or macrophages, and be expressed. And this way, they can induce quite robust immunity, both humoral and cellular. That's what people have found till now.
All right. Okay, let's see questions. Dr. Taufik Hidayat, our pediatrician, asks, how long will immunity persist after that 2 doses of mRNA vaccines? If any one of you can
Actually, nobody knows. Nobody knows. But expected that it will last at least 6 months to 1 year. At least 6 months to 1 year.
All right. Is there any other question? Okay. Zaki Zaili, I'm not sure who he is, asks, since the new approved vaccine require ultra cold environment, will this affect the effectiveness of the vaccine if the temperature are not maintained strictly? And what are the strategies to overcome this logistic issue? I think Dr. Hidayah, I think cold chain.
Okay, Dr. Zaki Zaili is actually a clinical microbiologist, one of my colleagues.
Okay.
Yes. Yeah, it is basically, it is quite critical to maintain the stability of this RNA, because RNA is very unstable and easily degraded. If the cold chain is not maintained properly, thus it will render the vaccine less efficacious. The cold chain starts from the manufacturing up until it is being given to the recipient. So this one, it should be maintained. So Pfizer vaccine in particular require an ultra cold storage condition. But a few newer generation vaccine try to go for the temperature that are stable enough, where you can just require a fridge to store it. Type of vaccine such as the AstraZeneca, I think is quite a stable vaccine which can be stored at a temperature of 2 to 8 degrees Celsius. So people should be looking more into this, because if you were to maintain an ultra cold condition to maintain the cold chain, it is very tedious. It might not reach the targeted population as you wish to reach lah.
Okay, I'm looking at the chat box now. We have a few more questions, but okay, for example, like, is there any, from Prof. Salam, is there any schedule to vaccinate persons already infected for COVID-19? Ada schedule tak vaccination COVID-19 now?
Those schedule, I haven't come across any schedule, but the subjects that are being studied are basically those who have not had any COVID-19 infection. The study do have data from those who are infected, but it is yet to be published lah.
Okay. Dr. Salam also asked whether there is any seroprevalence study done, but I think in Malaysia. So if anyone know about that, any seroprevalence study done about COVID-19 in Malaysia so far?
I think there's one study conducted in UMMC. I'm not sure about the exact seroprevalence, but it is in generally low. As you're getting away from waves of a COVID-19 outbreak, the numbers of those who have the baseline antibody towards COVID-19 is getting lower. As is expected, and because the antibody only can last to up to 6 months to 1 year at least.
Yeah, for seroprevalence study in many parts of the world, they discovered that the prevalence is rarely go beyond 5% from the population, mostly around 2 to 3% seroprevalence study. Those from all over the world, I'm not sure about Malaysia. Okay, we have question specifically for Dr. Hidayah. I hope it's not another of your colleague. Does the article mention anything particularly regarding the exposure of COVID-19 post-vaccination? I repeat, does the article, I'm not sure which article he's referring to, mention anything particularly regarding the exposure of COVID-19 post-vaccination? How does the 8 participants in the vaccine group and 192 participants in the placebo group have the COVID-19 onset?
They don't. I think that this is one of the pathology MO basically. I think the interim report, the report does not detail out how the 8 participants in the vaccine group acquired COVID-19. The report just mentioned how many.
Okay, so okay. And question from Nara K. Will there be any long-term side effect of the mRNA virus, mRNA vaccine, such as increased risk of infection by different strain of the virus? Prof. Imad, perhaps?
I think this is also a question, it's too early to answer this question, whether there will be on the long term any side effects or reinfections with new strains of the virus. As we mentioned before, non-neutralizing antibodies, if they persist and you get another infection with a virus that is not affected by the present immunity that you have, then there might be some reaction. But immunity usually does not last, as we said, long enough. It will wane off within probably 6 months to 1 year.
Yeah. So basically, there's many questions with regard to whether how actually people are querying how effective the vaccine is. So there's another question. I think it's already answered that the immunity won't last long. I think there are many vaccines that do not have a long life immunity. So even including vaccine, even just now, Prof. Imad mentioned that within 3 months, the antibody titer, the levels drop by half over 3 months time. Yeah. So I think that's answer for most of the questions. All right. Even about booster, when Prof. Salam mentioned, already mentioned in the presentation. I would like to ask, especially the general questions, the concern of the public. So basically, from your 2 presentation, I would say that at the moment, the short-term observation that we see, the vaccine that currently available is safe. That's what I can see from both of your presentation. For short-term, what we know so far, they are quite safe. There is no evidence to say that they poses any immediate hazard. But Dr. Hidayah did mention a case of the dengue vaccine that have a long-term 5 years, very adverse, very dangerous effect. The people die or maybe a severe dengue infection after that. That can happen. That's the Dr. Hidayah cautioned that this is still possible. But having the development of COVID-19 vaccine, especially when they suddenly able to develop so fast and they are not following the usual, because now Prof. Imad mentioned about accelerated development. I wonder, Prof. Imad, is there, when we suddenly allow accelerated development of vaccine, will that somehow make the vaccines less safe? Again, I know that currently we state that they are safe, but all this, so the need to have vaccine, somehow create another.
What has been published till now is that there is no cutting corners regarding the safety, especially the safety. But the safety has been noted only for this period of time, short period of time. We don't know on the long run. For the time that they have been tested, they seem to be safe according to the data.
So if we were to advise the public, so now public is very concerned. So we are, most of us here are professionals, are students in medicine or healthcare. We are interested looking at vaccine, and we have people outside. Maybe there are also non-doctors, non-health professionals listening to the presentation now. So what could be our advice to them with regard to the incoming vaccine, regardless from Pfizer, mRNA, or different type of vaccine that currently available? What is your general advice? Maybe I can start with Dr. Hidayah first.
Okay. I think I've think of how to answer the question last night, but somehow I forgot how to answer.
Okay. I mean.
Okay, Prof. Imad, answer dulu.
These vaccines.
How about you, Prof. Imad?
Yeah, I'm trying to answer. Can you hear me or not?
Yes.
Yeah, okay. Well, these vaccines, as we said, till now they are apparently safe, but there is no guarantee, of course. But the thing is that at the personal level, you might say, I would like to wait and see how the vaccine on the long term, how does it behave on the long term. But remember that we are facing an emergency. It is a pandemic. And if we wait, I mean, there will be more suffering and more deaths. So as a commitment to the community, we should take the vaccine. But personally, you might say, no, I can wait. I can take precautions and I can wait for some time.
Prof, for me personally, I would assess the current situation. If the surrounding transmission is very intense, is very high, I think I need to protect myself, I will take the vaccine.
Yeah. Because I'm public health, I would say that we should stratify who or prioritize who should be protected first. Maybe the healthcare, the frontliners should take the vaccine first, and then maybe the rest will be the community or those high risk, example like old people, like people with cardiovascular or those comorbid. So high risk people and those who, let's say, he got infection and can easily succumb to the disease. So maybe we can prioritize that. And there's also actually a questions asking the panel. Who do we have the schedule? Who will get the vaccine first? The answer actually there is no schedule. We do not know who, when the government obtain the vaccine, who will get first. We do not know. So somehow macam as if social media people want to know who will get first. So previously the one old people in UK got first, and then one lady nurse and collapsed during the press conference. So as if people want to know who will get first. So I don't think that should be our aim lah when it comes to vaccine. So okay.
At the national level, the discussion have been initiated lah with all the stakeholders involved, and government are moving towards vaccinating the frontliners first, and followed with all the high risk population, which is the patient.
In Malaysia, Dr. Hidayah mentioned we have an organization, our NPRA will look into the safety of any drugs coming into our country first. They will scrutinize whether it's really safe for us. Even though we commit to purchase, doesn't mean that we will accept. So the authority will further scrutinize and further monitor, inshallah. So the last, I would say the last remark from both of you. Start from Imad, Prof. Imad first. Your last remark for all of us.
Okay, I think you should take the vaccine when you can get it.
Okay.
Dr. Hidayah.
I'm decided to agree with Prof. Imad's statement. Inshallah.
All right. I think that's all from us. It's already
Okay.
okay. So on behalf of the kuliah, I would like to thank both of you, Prof. Imad and Dr. Hidayah, for the excellent discussion with regard to vaccination. I hope after this we are more knowledgeable. Thank you everyone who joined our discussion today. Thank you for the questions and all the best. And we make dua that this pandemic will over soon and we get our life back, hopefully. With that, I thank you everyone, and we end with tasbih kifara and surah Al-Asr. Subhanallah wa bihamdihi, subhanakallahumma wa bihamdika, asyhadu an la ilaha illa anta, astaghfiruka wa atubu ilaik. Wal 'asr innal insana lafi khusr illa alladhina amanu wa 'amilus shalihat wa tawasaw bil haqq wa tawasaw bis sabr. Assalamualaikum.
Wa alaikum assalam wa rahmatullah.